if you take a careful look of the animals around us, you might find something that seems to be common sense, actually quite interesting. Like these wild geese up the autumn sky, these monarch butterflies taking off from trees, or turtles in the ocean. They all seem to be heading somewhere, but where? Well, to their new home, of course, you might say. They're migrating for thousands of miles to a warmer place. A good one. But here we have a further question. How do they manage to find the exact same place needed to go up from hundreds of meters in the sky or under the surface of vast ocean, and keep doing the exact same thing for generations? You might frown and put your finger under your chin, only to find that you couldn't come up with a precise answer. Learning? Genetics? Or simply a pair of good eyes? Not exactly. In fact, scientists have been discussing this question for decades. Almost all possible hypotheses were already proposed and refuted. All but a promising one, the magnetic sensor protein theory. In order to introduce this theory to you guys, we must first talk about something necessary to understand it. You see, the Earth is surrounded by a round magnetic field. And we all know that magnets can be used to point out geographical direction for people. Therefore, numbers of scientists think there might be some kind of a magnet in the bird's brain to guide them with magnetic fields. They call this kind of idea ferrimagnetism. Well, others believe that because organisms is functioned by millions of chemical reactions, thus the nature of this magnetic guide should be also chemical reaction. Based on this kind of view, scientist Schouten proposed that cryptochromes, also known as Cry, might be the magnetic protein because it has occurred radical pair reactions. However, both of the theories have evidence supporting them and gaps that haven't been solved. Then there was a clever guy named Xie Can thought, why don't we combine these two theories together? With this, we got a theory claims that there is a magnetic receptor that is formed by Cry and polymers that contains iron. The magnetosensor complex should have a rod-like shape in order to better respond to the magnetic field. To prove the hypothesis, scientists tried to find the possible proteins in fruit flies. In the Drosophila gene group, there is 12,536 genes. Among them, they use computer to screen 98 of them, with conditions that it is in the head. It contains iron, but it isn't in the memory of the cell. Then, by using biology knowledge and experience, they focused on 14 proteins. The scientists found out that 9 of them are co-expressed with Cry in the head of the fly. Finally, one protein was found. The scientists named it MAGR. The second step is to find out the structure of Cry-MAGR to see if it accords with the hypothesis. By observing under the electronic microscope and homology modeling, the scientists proved that the structure was what they expected. The last step is to see whether the Cry-MAGR complex can respond to magnetic field or not. Scientists first count and record the number of complexes in different directions. They verify the magnetic force using Ferris powder. Then, they change direction of magnetic field to see whether the complex will change direction too. Finally, they use magnometer to test the magnetic field of the complex. Now, Dr. Xie and his team had proved that Cry-MAGR complex can respond to magnetic field. Yet, there's still some gaps in his research. Dr. Xie had reported that the complex would only contain 40 iron atoms, but Dr. Meister argued that the smallest known naturally occurring iron-based magnet has 1 million atoms packed in a small space. At the same time, they propose that there is no evidence of a direct interaction between the magnetic induction proteins and Cry molecules. It is pointed out that the Cry4 sequence used in this paper is wrong. So, if you want to determine whether it is magnetic induction proteins that can navigate animals, the first thing you need to overcome are these problems. Therefore, we propose some further questions here. Mag-R responds to ordinary magnets, and in theory, to the magnetic field of Earth. From the perspective of magnetic induction elements, how to make the protein have higher magnetic sensitivity, and how to use the Mag-R protein to convert magnetic signals to biological signals still need further exploration. Here we give our own hypothesis. Due to the unique magnetic properties of Cry-Mag-R complex, it may lead to electromagnetic induction by magnetic fields based on Mag-R protein and the Earth. The voltage it produced may be transported by neurons as neurosignals. Anyway, this is just our guess. To discover the truth, we need substantial experiment data and exact analysis. Modern science is established on these things.